Today's topic is plant regeneration pathway. When we do plant tissue culture, we take explant, surface sterilization, inoculation on a sterilized medium, then we are allowed to grow in the growth chamber. Now the question comes, we get a group of tissue in the growing chamber on a sterilized medium. From this we have to regenerate that plant. The regeneration of plants from this tissue is done by two ways. The first way is organogenesis. The second way is somatic embryogenesis. We'll discuss these two pathways in detail today. Here we can see citrus plants. We regenerate citrus plants from the explant through plant tissue culture. Here what we do, first we generate seedlings. If you see here, they are generating the seedlings by germinating the citrus seeds on a nutrient medium. And we transfer that epicotyle to a medium. And first we induce shoot from that. Then these shoots separate and keep on a root medium and generating root. Then they are kept for hardening and transferred to the field. So if we take the sequence here, first we take explant, then in the second step we start growing them, and in the third step we generate shoot, in the fourth step we generate root, and in the fifth step we generate plantlets and we go for hardening. So the method used here to regenerate plant is through organogenesis. See this explant, we generate callus from that. The callus can be induced for shoot generation, and the callus can also be induced for shoot generation. After shooting and rooting, the plantlets will be formed. So the approach in both these figures shows that we generate roots and shoots and then go for producing plantlets. This method is known as organogenesis. Next method is somatic embryogenesis. See the word very clearly, it is somatic embryos. We take the explant and they are kept on a medium to generate callus. No doubt, based on the oxygens that you give, they will generate callus. Callus is induced for formation of embryo. We are giving hormones. We are introducing the cells of callus to a particular medium where the callus cells will induce to form embryos. The different structures of embryo, they will form a group of cells, elongated cells, and develop torpedo-like structures. These torpedo-like structures are actually shown here, and they will be allowed to grow in the medium. In the torpedo-like structures, you can see the plumule and radicle. They are the initial root and shoot. Just we need to grow these embryos in a suitable medium. They will generate the plants, and that plants can be kept for hardening. From the callus, what we do, we go for somatic embryos, generate embryo from that. Now the embryo is allowed to grow. Here we are not going for shooting and rooting. Here we are going, individual cells are allowed to grow as embryos. Method of regeneration is somatic embryogenesis. Now we will see both together. We take explants and we generate callus from that. From the callus, you can go for root and shoot formation. Then those plants will be allowed to grow as plantlets. This is indirect organogenesis. That means we generate callus first. Otherwise, if you take shoot tip आपके पास है तो उसमें बस root generate करना है. Vice versa, अगर आपके पास root के portion है तो you have to generate shoots from that. Those are direct organogenesis. But these two methods are organogenesis. Whereas in the other method is direct, we generate embryo from that. That is direct somatic embryogenesis. Otherwise, we'll go for callus. From the callus, we'll induce somatic embryos. And these embryos are allowed to grow. The embryos are somatic embryogenous embryos because they are not formed by the fusion of male and female gametes. They are generated from somatic cells. This pathway is known as somatic embryogenesis. So these are the two pathways of regeneration of plantlets through plant tissue culture.